Good evening, everyone. We, the members of Biology Skill Development Forum, cordially invite you to the second session of the Journal Club. Today, we are fortunate enough to host Ms. Susmita Ghosh, who is going to speak on the topic Nanoproteomics, analyzing the microcosm of biomolecules in the nanogram range. Ms. Ghosh is currently pursuing her doctoral studies from Leibniz Institute for Analytische Wissenschaften, ISS, Dortmund, Germany, as a member of the Biofluorescence Research Group. Ms. Ghosh completed her bachelor's degree from Calcutta University, followed by master's at Utkal University, Odisha. After a short stint at IIT Bombay as a GRF fellow, she moved to Germany for her doctoral studies. Now I cordially invite her to begin the proceedings for the day. Meanwhile, I will request the audience to post questions for her in the chat, which she can handle at the end of the session. Thank you. You can start.
Yeah, thank you for the kind introduction. Just let me share my screen. It's visible, right?
Yes, it's visible.
Yeah, okay. So, good afternoon or evening, everyone, and thank you for the invitation here. Um, today I will mainly talk about the nanoproteomics. Basically, I will explain why nanoproteomics is necessary and what are the challenges in this field we are facing currently, and also we will see if we can find ways or the recommendations that scientists are given to find out something from this empty tube to get some this volcano of proteins. So the nanoproteomics, the terminology itself is very self-explanatory. Basically, when the nanotechnology meets proteomics, we call it nanoproteomics. Now, why nanotechnology is required here? Because when we really want to analyze the material down to nanogram range, at that level, we need to miniaturize the current technologies available. For example, traditionally or usually we analyze the samples which are at microgram or milligram level or such a bulk sample. But when we have cells or amount which are at 500 nanogram or even at picogram level, at that time we need to miniaturize the current proteomics technologies, and those are basically termed as nanoproteomics. Oh, sorry. Yeah. Now, why this nanoproteomics matter? So, why we are here today or talking about the nanoproteomics? So, we in our scientific research we are not always analyzing the bulk cells or cell lines, but sometimes we are also interested in single organ, for example islets of Langerhans, or also subcellular structure, for example the endoplasmic reticulum, or maybe some vesicles. In that case, if we want to do the proteomic characterization of this sample type, we need techniques that can handle such a minute amount without any losses. And the main promise behind this application is, for example, you know about the single cell proteomics, I think, that we can use it to understand even within the same cell type how many subtypes are there, or how one cell is different from the other cell, even within a similar type cell types. And also, if we have some kind of rare cell type, we can characterize them based on their surface proteins, what are the surface markers present, also the structural characteristics one can get, as well as the functional characteristics. We can know about the entire proteome of those rare cell types. And also, when at clinics we are not always get a lot of biopsy samples, right? So sometimes we get the fine needle samples of this tumor or maybe any tissue, so we can characterize them even when we have such a limited samples. And if we see just the history or the journey of single cell proteomics that we have right now since last 14 or 15 years, it's emerging quite nicely. And currently, we can say that this is the field or the hot topic in the proteomics field. So most of at the beginning, it's always started with the cell line like HeLa or MCF7, the breast cancer cell line. But these cell lines are they always have a bag of proteins in most of the cases. It's challenging when we want to replicate the same procedure in actual clinical samples, but nowadays also people are jumping into that area. They are either analyzing the big cells, for example the osteoclast, or they are doing LCM, like laser capture microdissection of the tissue. So this is how the journey has begun, and so far, and it's also emerging now, not only in the proteomics but also in other omics technologies like metabolomics and lipidomics. Now, the question is, what is the major problem? If we look into the central dogma from gene to protein, if we have the scarcity of the sample, then in each stages, if we want to analyze that sample, that is a problem. Then why the hype is only with the nanoproteomics but not with nanogenomics or nanotranscriptomics? The reason, or we can say, like, we are thankful to the PCR reaction, like polymerase chain reaction, because we can amplify the RNA or the DNA as much as possible, right? But for the proteins, no such amplification is possible. One can induce the protein expression or enhance the protein expression, but that cannot be like exactly replicated as it is possible for the RNA and DNA, because in case of protein expression, we know that it's not only just the expression, but also they have the folding and modifications. That contributes to its functionality. So that is the main problem in the proteomics field, that we cannot amplify, and that's why we really need to find out ways that we can deal with such a minute amount. So, what are the technical challenges then we are facing in this field? So, basically, whenever we want to do proteomics, we try to get as much as proteins from the sample available. If we look into the bottom-up proteomics workflow, then we first isolate them, then prepare the sample for the proteomics, and then proceed for the separation or using the chromatographic methods, followed by mass spectrometry. And in each step, we face problems. For example, we lose sample while preparing them, and it's crucial when we have the minute amount. Then the separation of the sample through the chromatography, that is also a major point here. And at the end is the sensitivity or accuracy, how accurately we can identify and quantify our proteins from such a minute amount. So today I will mostly talk about this first part, the sample loss during the preparation, but I will also touch upon this two part later, but not in a very depth. So in bottom-up proteomics workflow, if we look into the sample preparation, these are the four major steps. First, we isolate our cells or tissue from the body, and then we isolate the cells from the body fluid or tissue, and then we lyse them to extract the proteins and remove the debris. And third point, we digest them to break into small peptides, and further we need to clean them up before the MS analysis. And we basically lose samples at each step. So, what are the major reasons for losing samples, or the common reasons in losing samples in all these steps are tube to tube transfer. So, whenever we prepare the samples for the lysis followed by digestion, we have a tendency that we transfer into fresh tube. And each time we transfer, we lose something. It's not only because of the pipetting volume or our pipetting error, but also because of the phenomenon called or peptide adsorption by the tubes. So the tube, the Eppendorf tube we are using, majorly using Eppendorf tubes, they have a tendency to bind to this protein and peptides. So each time we transfer, each time a part of them are bound to the tube surface, and then we lose our samples there. So that's why the major recommendation for working with this minute amount is one pot sample preparation. So you lyse and you digest in one tube and then proceed for further LCMS analysis. But what are the other problems we face? For example, when we isolate the cell sorting, which is very commonly known, specifically this fluorescent activated cell sorting, where we basically tag a cell with the fluorescence and we isolate that target cells from a mixture of cell population, and we sort them separately. And during this sorting, we use a laser to scatter or to check their fluorescence scattering. While we use this kind of charge or laser, that cause a stress to the cells, and then the cell can be died. For example, the neutrophils, which is one of the common immune cells, they are very sensitive this kind of laser or the charge. So if we are handling like million cells, then at that time it's not a problem. But when we are handling very minute cells, for example 40 cells, 30 cells, at that time if we lose half of them, it's a real trouble. So we need to find out a way where we can sort the cells without giving such a stress. And also, probably many of you know that when we do the cell culturing and after that we have to wash the media from the cell, we wash them with PBS. And at each step we lose some cells, and this is very crucial when we have the minute amount. So definitely there are also other major problems, but not all of them can be addressed at the same time. But this company called Cynion, they try to address as much as possible, at least the one that is caused by the manual handling. So this company, they have designed one instrument, we can call it, which can isolate the cell in with high accuracy in an automated manner, and also it can handle a very minute volume, for example, at picoliter level. So this is a short video, which, yeah, it's working. So here you can see that we sort the cells in a plate. So they have either 96 or 384 or 1596 well plates. They also have a temperature control within this instrument so that it prevents RNA or protein degradation. So once have a mixture of cells, so they take out the cells within pipette and they have a camera in front of that. And or the microscope, we can say. So this microscope, it can be either image-based microscope or the fluorescent-based microscope. So they have two areas, one ejection area, another is the sedimentation area. And when the cells are there in the ejection area, they detect it. For example, now they could not detect any cells. But when they detect one cell, they transfer that cell to one well. And then they come back again in front of the camera. So the number of times they detect one single cell, they dispense that single cell to each well. And when there is no cell, they just dispense the liquid. But sometimes they also detect multiple cells. That is possible. That is also going to waste because system is very accurate for single cell analysis. Now the question will be how they detect single cell. It's based on different properties of the cells like diameter, elongation, and the circularity. So this kind of information is stored there, and you can give an instruction to the instrument. Okay, I want to isolate the cells within this diameter, and this kind of debris are also then discarded. So it can isolate up to 40 single cells in each minute. So now you can imagine that we don't have to do any give any stress to the cells. Also, we don't have to just pipe it at a, I mean, it's not possible to pipe it such a picoliter level, right? So this is how the cell worked, but also it's a problem with the tissue, because we often cut the tissue manually, and we know when we cut the tissue at that time, either we lose some area, or many problems can happen during cutting the tissues, right? So there's a technique called laser capture microdissection, where we can for example, if we have a liver tissue and we only want to cut out the necrotic part, which is very minute, at that time it's not possible with the manual cutting. So in that case, the laser capture microdissection can help, where we can define the region and then it can dissect that exactly that section, and you can further proceed for the LCMS analysis. So after the isolation, the next challenge is the lysis. Definitely, the major question is which method we should use for the efficient lysis of the cell and solubilization of the protein. So it's very known, like commonly we are using different mechanical method, for example, the sonication or also the motor pestle that many of we already use, but it depends on which sample type you have. For example, if we have some solid tissue, then we prefer to use this motor pestle. But if we have some kind of cells, then even freeze-thaw works very nicely. You don't have to use any other apparatus. But also sonication is kind of the one standard I mean standard mechanical methods that we use in every proteomics sample preparation technique. But not all of them are 100% efficient in dissolving the proteins, specifically the membrane proteins, because membrane proteins, they really need to soluble they need to be solubilized from the lipid fraction. And in that case, we prefer to use the detergents like sodium dodecyl sulfate, or SDS is commonly used detergent, but also people use others like NP40, Triton X, Twin 20. These are strong to mild lysis agent, and also they some are non-denaturing agent, which are very good for and efficient for lysing the cell and also for the protein solubilization. But we need to remove this detergent before mass spec analysis. Why? Otherwise, you will see in between your nice chromatogram, you will see a garbage of this detergents. How it exactly looks on the chromatogram is this. So when you see then such a nice chromatogram, you are very happy because you're sure that your digestion works perfectly and you can see some peaks. But then at the middle, if this is the SDS, which ruin the entire analysis, and not only your sample, yeah, I mean, you lost your sample, but not only that, but also the mass spectrometer is also gone. So therefore, we really need to be careful that we remove this kind of detergents prior to mass spec analysis. But there are some common detergents as well as some electrical methods used for such a minute amount, which I mean, the list is really long. But what is commonly recommended is if we have a combination of the sonication, where we don't use the ultrasonic probe, but it's just a bead-based sonication or just a water bath sonication, and a detergent that is mass spec compatible, so that we don't have to worry about removing this compound or the detergent from the sample. Then we skip at least one step of this cleanup, right? And we don't, the less we touch our sample, the less we are losing. And also the freeze thaw cycle, there we don't need to use any like mechanical other mechanical step or the sonicator or even the detergents. And the single pot preparation. So here is one paper who, I mean, this group, they nicely shown that they use DDM or maltoside. This is also a surfactant, and it's a very effective in solubilizing the membrane proteins as well as other proteins, and it's very compatible. So they did this one pot sample processing. So after sorting the cells or cutting out the tissue section from using the laser capture microdissection, they processed in the one tube. They performed the lysis for the digestion and finally submitted to MS analysis. And here you can see from just 20 cells, they were able to identify like 1200 protein, which is a lot. So overall, from here, I think the major key point is we need to think about using a MS compatible reagent as well as one pot sample preparation. The next two tips are quite correlated, the digestion and the sample cleanup, because we need to clean up our sample even before digestion. For example, if we have SDS in our sample, it can hamper the digestion because for the digestion of the protein into peptide, we also use some enzyme. And also after the digestion, we need to remove the contaminants. Otherwise, I have already shown you how it can hamper the MS analysis. And the digestion conditions, like which enzyme we are using and what is the ratio of that enzyme to protein. In mass spec, we are mostly using trypsin. It's a serine protease which cleaves the peptides at a C terminal of lysine and arginine. And this is the structural or the catalytic triad of the trypsin. It has been shown that even within the trypsin, so it's an enzyme, but at the same time, it's also a protein. So it can digest itself as well. So the companies, they have tried to develop or try to synthesize a protein which has more resistance to autolytic digestion. So that's why now there are different types of the trypsin, like trypsin platin or trypsin gold. So this kind of trypsin you can see here, when which has more resistance to the autolytic digestion, it gives more protein coverage. And also some enhancer, like because we know that enzyme needs a cofactor, and also sometimes some optimal condition we can use to induce or to promote the enzyme activity. And here, this group have also shown that using a surfactant, it induces the protein or the protein coverage by 17%. So this one can keep in mind before selecting which enzyme they can choose and whether they can use extra cofactor or any other enhancer there to promote the trypsin digestion within such a short span of time, incubation time. So usually when we digest the protein and we clean them up, we follow this two strategy. Like in most of the cases, one will be the gel-based strategies, and another is the in-solution strategies. So in case of gel-based, basically we have the protein and we run them on gel. We cut out the gel band and then we digest the proteins further, and then we have the clip peptides, which we measure on LCMS. On the other hand, if for in solution, we have after the lysis everything in there in the tube in the solution format. You just have to do the digestion, and you then once we have the peptides, you just have to clean up the sample and further perform the LCMS analysis. And during this cleaning up, there are various strategies available which can combine both this digestion as well as the cleanup procedure. For the low cell number, as I said, like multiple time we process the or we touch the cells, we lose them a lot. So for the low cell number, there are some different strategies people or the scientists have found that can reduce the loss as much as possible. So one is the bead based, where basically the protein bind to the beads or to the magnetic beads, and one can just remove this solvents which has all the contaminants like salts or SDS, this kind of stuff. Another has a stage tip based, which is nothing but a filter, and also the fast reactor. This is also a filter based. Basically, we pass the protein through the filter, and then we do the digestion on the filter, where only we have proteins and the enzymes, and all other contaminants are passed through. And another, or which is I would say very nowadays a bit common for single cell proteomics, is the nanopods. So here we have a chip, and in one chip represents basically one well, and in one well you do all the reaction like adding sample, doing lysis, and then digestion, and further from the same well it take up the sample for the LCMS analysis. So there at ESAS, what I am doing is I'm also working with a minute amount, not with a single cell, but with a amount which is at nanogram level. So I'm using a filter. It's known as the S-trap filter. So I have the protein in the SDS lysis buffer, and I pass through this sample using a filter, using this filter, and then proteins are trapped onto the filter, whereas the other contaminants are passed through it. And further, I wash it with the buffer, and then do the trypsin based in the digestion of the protein onto the filter, and then finally elute the peptides. And I have modified this protocol at ESAs at each almost in most of the steps, but I cannot show you the parameter right now, as this is not available publicly yet. So you'll have to like keep an eye on it later maybe for the publication. But what I found is using this modification, it induces or it increased the protein coverage. As you can see, this one was the traditional or the company based method, and this is the modified one by me. And I already got like around 400 plus proteins. And not, I mean, this is just for 200 nanogram, and also from 50 nanogram I got a really good coverage. So overall, I would say there are very different type of publications nowadays are there, and different type of approaches are there that one can use for to handle such a minute amount of sample. But it depends on now the question, like which method one should choose. It depends on the sample type you have. As I say, the HeLa is a bag of protein, so even like every method works for HeLa. But for own sample type, we need to figure out whether this method is suitable or not. Second is which biological question we want to address, whether we want to just focus on the membrane proteins, then we have to select that kind of detergent for the lysis, or we want just a global proteome and we are not interested mainly on the membrane proteins. At that time, we can also deal with such minute or mild lysis agent. And how much is available? Because some of the techniques which are very fancy, it's good, but is are also expensive. So we need to think beforehand, like whether really we should use this method for the material we have, or maybe the technologies that are already available that are cheaper, we can modify just a slight and we can use it for our sample. And also some of the single cell proteomics methods are not applicable when we have like such a huge amount. Yeah, I think I have covered a bit of this sample loss that we face during the preparation and what steps one need to take care of while preparing the sample. The second two steps are the separation power, the throughput, like the chromatography, and the mass spec, the sensitivity of the mass spec. So the chromatographic separation, if we, I think many of you already know about the principle of the chromatography. So basically here we use a column and our molecules and peptides, we pass through this column, they bind to the column first, and then we elute them with solvents, mainly the non-polar solvents. And this is how one HPLC, not how HPLC looks like, but if we think about the schematic diagram, this is how it will be in a more simplified version. So we have a mobile phase, which is nothing but non-polar solvent, and we have a solvent delivery pump here that pump our mobile phase through the column. And then we inject our sample here, which then binds to the stationary phase of the column and separates when we pass the mobile phase solvents. And finally, it's detected using the detector, and you can see this kind of graphs or the peaks. And yeah, for the illusion, what we do, we basically can decide the gradient or how we want to elute the sample. So the non-polars, we use a particular gradient of the non-polar solvent to elute this kind of peptides or the molecules. So here maybe this will clear a bit. So here you can see we had first, sorry, it's a bit faster. So we had basically a mixture of the peptides, and yeah, here we have a mixture of the peptides, and when we pass the solvents or the non-polar solvents, then it's separating, and each time the each type of group is separating and we are getting a single peak. So this is the basic principle, or this is how we are getting the peaks of the UV chromatogram after the HPLC. So what are the factors then we need to optimize if we want to increase our sensitivity for the chromatographic separation? Is column material. This column, it's made up of resin. So, and each resin has different property. So depending on which kind of sample type or which kind of peptides you want to elute or you are interested in, you can decide the column material, or also whether I mean, it also depends on not only in the proteomics but also in the lipids or metabolites as well. Second is the column diameter. So this you can see if we have very minute amount of sample, at that time it's preferred to use a reduced diameter, because if the diameter is too big, then it's tend to lose everywhere, or like one can say like diffuse to everywhere. And it's same for the column length. So when we have really a minute amount, it's preferred to use a reduced diameter as well as a reduced column length. And also the flow of the solvents, like how fast you want to analyze or you want to separate in a your analytes in within a short time period or with a fast time period or a long time period. So the time, the flow of the solvents, the length, diameter, all this matters here for to separate the analytes when you have a really minute amount of peptides. Here is an example. You can see when we use 75 micron inner diameter of the column versus when we use such a 30 micron inner diameter column. With reduced diameter size, we get more intense peaks than this one. And this happen only when we have such a low amount. If we have high, then of course we will prefer to use the bigger inner diameter LC column. The last stage is the MS data acquisition. In probably in a previous lecture you have already got a very nice overview of this mass spectrometry, the proteomics, and about the label based, label free techniques. So here I'm not going into more details of that, but briefly. So when we have the peptides, we first separate using the chromatography, and after that it goes to the through the ion source. It goes to the analyzer, and further one type of analyte is fragmented, or the ion is fragmented in the within the collision cell, and further it's analyzed in the second analyzer, and then we see the MSMS spectra depending on their mass by charge ratio. So what are the factors there that also contribute or one need to consider in this minute amount or the nanoproteomics field are the analyzer type, which analyzer we are choosing for the better sensitivity, whether we want a high resolution, low resolution, that also depends. Second is the data acquisition mode. So in proteomics, we are using basically two different data acquisition mode. One is called DDA and another is called DIA. In DDA, so basically you can imagine in the first analyzer or the analyzer one, you are getting a lot of peptides like this, and it only selects this high abundant one and further take forward for the collision self based fragmentation, and then you only in the analyzer 2 only these high abundant peptides are selected or their mass by charge ratio you can see. Whereas in the DIA, it does not depend on the abundance, but rather there we define the window. Okay, take all from within this window. We don't like discriminate, or it doesn't matter whether it's abundant or less abundant. So take out all we have within this window, and then proceed for the fragmentation and for the MS analysis. So that's why you can see more in the DIA. But the data here in DIA is more complex, which also needs a spectral library to know, like, okay, to validate whether the spectra is actual or not. It's just a false noise. The another dimension here is the mobility. Mobility. So we first separate our molecules based on the in the chromatographic separation, and then further we proceed to the for the mass analysis or M over Z analysis. But here nowadays, like people are introducing another dimension before the MS analysis, that is the mobility separation. So it means the peptides which are first separated on liquid chromatography, further separated on the based on their ion mobility or mobility, which is basically a function of their shape. As you can see here, the one which is bigger is going faster than the others. So, so it separates the molecule based on their mobility, and then it goes to the analyzer. So in this field, right now, two mass spectrometer are kind of competing. One, it's the orbit trap, which is from the thermoscientific, and another is a timsTOF SFP from Bruker. So orbit trap here, this is how the mass analyzer look like. So basically, the ions are trapped within this electrodes, and it measured the m over z based on the frequency of the oscillation within a time. And here it in the timsTOF, it basically in the TOF analyzer, it measures like the within a time, like an object travel a distance through the medium like this. So both of them are quite good. Like they have different mass resolution and mobility resolution, but they have really good sensitivity. So it's not decided who is getting the best prize yet. So overall, the in the nanoproteomics, the things that we need to consider, from sample preparation to chromatographic separation, data acquisition, and applications, are first we need to think about, we have to reduce the loss. Second, we have to be fast and reproducible. Of course, fast, otherwise it can also take time and also evaporate the samples. And reproducibility, this is a major thing in the proteomics. It makes no sense if you have no reproducibility among the biological replicates, then you will obviously see a variation in your data and cannot say whether this variation is from the biological reason or from the technical preparation. And in the chromatographic separation, yes, we have to think about how we can improve the separation power, the longevity, and again the reproducibility. So longevity is mainly due to the column, like which kind of column material or how we can use it so that it can last long. And the data acquisition, it's also about the quantification, accuracy, and speed of the mass spectrometer to scan this kind of data, and how sensitive one mass spectrometer is to acquire as much as possible. What is the resolution, and how many proteins you can accurately quantify from such a minute amount. And of course, with all this, you can apply and address different biological questions, which includes the tumorigenesis, like you can apply to know about how one cells are different from the others, like cellular heterogeneity, as well as within the tumor microenvironment, what are the different cell types are there and how their behavior and all. So to this, I actually uses different resources, and if you want, I can share this with you, or also I think you can get it from the video. So the few papers are really good where they have very detailed with detailed description about how or what factors one should consider for handling such a minute amount. And with that, I would like to thank you for your kind attention. I think I'm a bit finishing a bit early, but I would like to spend time in discussion if you have any questions.
Madam, hello.
Yeah, hi.
Hi, I am Shomia. Usually, for nano, what you're talking, does
Mhm.
it the column size or column length does changes? Are we using same columns what we use for arbitrap or general?
So column diameter, yes, you can change. As I showed already with reduced column diameter, it actually helps better when you are dealing with such a minute amount like nanogram range or even at picogram range. column length is also matters. Like usually, I think in the proteomics you are using a 25 centimeter or 50 centimeter sometimes, but it has been shown that you with 15 centimeter it improves the protein identification. The main reason is the diffusion. If you have a longer column and less amount, then it tend to like diffuse while traveling through the column. So if you have
Okay.
a reduced one, then it's faster and accurate and less diffusion happens.
But but that means the less is the internal diameter, the more the data separation proper and intensity you're talking. Am I right, madam?
Yes, yes. When you have a
And
minute amount.
I'm sorry to interrupt you. And
Hmm.
also, usually because you may be doing a thesis about all this work, in general, which like what diameter in standard you're using, madam, for this kind of work where you do a nano LC
Yeah,
or what is it,
so
nano?
nanoproteomics, that's correct. So when we are using the single cell or very minute amount, then we prefer to use 30 micron in a diameter column. Yeah,
13 micron. Okay. Okay.
30, 30, 30. Yeah,
Okay. Oh, understood. Thank you. Thanks, madam.
yeah. I can see one question in the chat whether these methods are capable of dealing with low abundant proteins. Yes, for the low abundant proteins, I mean, maybe you can explain a bit like which methods do you mean, whether it's the mass spec or it's the chromatographic separation or the sample preparation. So if I talk about the mass spec, then yes, if you use the timsTOF, it's actually capable of identify the low abundant proteins there because of this mobility separation. In the orbit trap, it's also possible if we use the frames in front of orbit traps for this mobility separation. Then it's possible.
Sushmita, actually, my question was that whether if you are taking only few cells, starting maybe 12, 15 cells after sorter, whether that
Mhm.
particular protein concentration what we are starting with will be able to even identify the low abundant proteins, or it will be only the 1200 or 1500 proteins that you are identifying. Those
Mhm.
proteins are only the high abundant or somewhere in the coming in between, but the low abundant protein sometimes which could be also a biomarker we are losing.
Mhm. Yeah, I mean, yeah, that is possible. Like with such low cell number, if we are starting with 10 or 12, it's possible that we may lose some. But so far, or the way that field is emerging, I would say we can still identify. Like I can share my experience. I have worked with so far 200 cells. And the most low abundant proteins we can now think about are the transcription factors mostly. And we are still able to see them even from 200 cells.
Okay, that means it's possible, right?
Yes. Yeah, it's
Yeah.
it's
Okay.
possible. But yeah.
Okay. Thank you.
So there is another question in the chat. So on which solution are you reconstituting the final sample before submitting to mass spec?
Well, yeah, this is, I think, a debatable question. But yes, one can use, we are using currently 0.1% TFA, but one can also use DMSO or just 0.1% FA. But we are using here 0.1% TFA. Is 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 that answers your question?
Hello,
So,
madam.
uh,
Yeah.
Yeah, and also I was asking that for the cell sorting,
Mhm.
whatever Selen one we are using, can we I'm just asking out of an experimental designing, and I'm asking. So have you tried with fax machine or any what is it, water and oil emulsion kind of a kind of that are we using anywhere? I'm just like, because I'm from little different background. Uh, just I'm asking. It is
Yeah. Sorry, I don't know if I get your questions correctly. So you are asking whether I used fax or?
facts or, and there is a one more what is it, instrumentation called water and oil emulsion like concept they use for molecule separations in molecular biology.
Yeah.
Is being used here? Like, I'm just asking. It is. I know it is a very naive question. I'm also very naive in the domain. Just I'm asking. Yeah.
No. That's completely fine. So the thing is, for the fax or whatever the Selen one, the technique they are using, it has pre-separation technique that they don't mention here. So you cannot just inject the blood onto the system and it can isolate the cells. No, that's not possible. From the blood you have to first isolate the cells and then you can proceed. This is the first thing. Second thing, no, I have not used the water oil emulsion, but I have used fax for my sample, as I just maybe mentioned once that I am working with immune cells. It's called neutrophils, and I am isolating these cells from blood or brain or even from spinal cord, and I am using two different techniques. One it's called the fluorescent activated cell sorting, and another is the magnet activated cell sorting. I personally did not use the Selen one, the one that I showed here. I mean, I still have the plan, but I have not used it yet. But I used fax and the magnet activated cell sorting. For me, the magnet activated cell sorting actually works quite better than the fax, because, as I mentioned, in fax, you are giving a lot of stress to the cells. And when you get actually, if you are aiming to isolate 1000, at the end, maybe you will end up at 100 or 200 cells sometimes. That's possible. Live cells I'm talking about. So most of them are dead, they are ruptured, so you don't know them. That then the counting and everything is a bit messed up there. So
In fact, the protocol is
yeah.
somewhere maybe little harsh. You should be, handling your hand should be very, very gentle, and the way you handle also it should be very gentle. If you don't
Yeah.
have that finesse, it will be very difficult to get what you wanted. Yes, I
Yeah,
understand.
exactly. And for the neutrophils, I mean the cell type I am using, it's very sensitive. Like even if you wash 3 times with PBS, it will change its post-translational modifications. Plus, also you will lose a lot of proteins. Sorry.
Thank you.
Yeah, you're welcome.
Another question in the chat is
Yeah.
that since you are getting less amount of protein here, how are you checking the quality of the samples as HDS is possible, is not possible?
Yeah, this is, I think, the problem I faced first time when I started with it. Yeah, this is a very good question. Here we use one standalone HPLC. It's called Monolith, and this monolithic HPLC also has capability to detect at least till 50 nanogram. So I have the amount I have worked with, or the starting amount I have worked with, mainly 50 nanogram. For that I have checked using the HPLC. The one below 50 nanogram, it's hard. We just directly inject. I mean, it's a kind of a blind thing that we directly inject to our mass spec. We believe everything is working, but also when I prepare for the 200 cells, where you can expect even um, how much, like 10 nanogram or so. In that case, I always prepare one blank. and that blank I inject on monolith, and I check if I can see some like contaminants are there or some crazy peaks. If I find, then of course I know the entire batch is, maybe there are some contaminants still there. I need to clean them again. Otherwise, I just proceed with the MS analysis.
And ma'am, to add on to this, have you
Mhm.
explored Perkin and Ilmer solutions for protein quality checking chips and all?
No, because I think in many cases the what should I say, like there the amount is a bit limited. I'm not sure about this case, but yeah, in many cases the amount is limited. For example, even if you cannot do the estimation assay, like you don't know how much you are getting or how you can decide the trypsin. So the idea for this, what we do here is we do some kind of amino acid analysis. So, I mean, we just lyse our cells with the HCl or the acid, and then we check the amino acid, and we have a already prepared calibration curve. And based on that, we check the amount of the protein or peptide present in our sample. So even quantity or the protein amount estimation is also tricky when you have such a minute amount. So I'm not sure. I have not used any other techniques, or at least I don't know if there are any other techniques for quality checking of such a minute amount.
Yes, yes, I understand, ma'am. Usually, because this Perkin and Elmer works with, I heard it works with a nanogram for quality analysis and all. If you
Mm-hmm.
still find, may use that, but I myself not used, but I have got through one of the, like, I use it to have that one for DNA and RNA quality checking and all. But when I had a, like, proper technical demo, they also showed protein chips also.
Yeah.
Maybe it
Are
may
you
help.
are you talking about the nanodrop one?
Nanodrop, no, not nanodrop. Have
Okay.
you with a type station kind of a thing or kayak cells where we check a DNA, RNA quality?
Mm-hmm. Mm-hmm.
Including
Yeah, it could be possible, but
maybe you,
I'm not sure if it's possible for the protein, because to my knowledge so far there are no such techniques for minute amounts. At least that's what I know. I mean, you definitely cannot afford the entire sample just to check the quality, right?
and also yes, that reason I was also asking is like how do you optimize this whole experiment if you don't know what your quality or how exactly, madam, how you, what is it, on what basis you would like to go for like optimization के लिए?
Uh, s- like what do you mean?
Like when we don't know like what kind of quality or whether sample is gone or not also,
Mm-hmm.
uh, how do you, what is it, come like decide whether.
Yeah. Okay. Now I got your point. So what we usually do for that case? So you have to separate your experiment in that way, like whether you want to check for the sample preparation or you want to just check for the data acquisition and the um, and the like mass spec or chromatographic separation. When first we optimize for the chromatographic separation, we prefer to use the minute amount from the bulk, so that we know that it's reproducible. There is no technical differences or no preparation differences, right? So from the bulk amount, we inject a minute amount multiple times, and we optimize the parameter based on that. For the sample preparation optimization, of course, you have just one shot, and that you have to, I would say, compromise there.
So there is another question in the chat.
Yeah.
For the LC part, do you use non-polar solvent or polar solvent to elute the peptides?
Yeah. So I can see it. For the LC part is as you said, yes, Yeah, that's also a good question. Yes, we use here the acetonitrile, 84% acetonitrile we use. Yeah.
Uh, Hi. So I think the question that's mostly over from their end.
Mhm.
So as most of the students who are there, they're mainly of the masters and bachelors degree and also from the GRA. So there may be one question that this is out of the track question that how to apply for doctoral positions in Germany or ISIS in general.
Mhm.
So if you
Mhm.
can share some thoughts on that.
Yes. So, I mean, this is also good for the master students and bachelor students that not only for the doctoral position, but you can also apply for the internship. We already had interns here before from BHU or this kind of universities, and we will also soon have interns from the Hyderabad. So for this you don't have to do the you have to go through all the comparative exams here. You just have to write PI or like supervisor. You have to write them depending on what is your interest or whether you are interested in this particular topic, and then you just have to write them with in an email, and that actually works here much better. For the PhD, I would say although they are check for the English fluency, but it's not very mandatory here. So the all the exam like GRE, TOEFL, it's not mandatory in Germany. But how it works here basically through the supervisor. So first you have to approach the supervisor. If your supervisor gives a position, then you are enrolled at the university. So, if you are really interested in a particular area and within Germany, then I would say like you can start searching like the positions in Germany in this area, and then you will mostly see the institute-wise. Advertisement, or if you are really interested in any scientist work, you can directly write them. That is also possible here. So you directly write them and then you get answer from them. And yeah, if any of you are interested in internship, because we also, as I said, we also take interns here, you can write me as well if possible. Then we can talk about this, or I can talk to my supervisor here. If there are positions available, we can take it.
Ma'am,
Yeah,
can you please
about
share
the yeah, sorry.
email ID, madam? Your email ID, kindly can you share?
Yes, I can write it in the chat. And one moment. Yeah, this is my email ID. And yeah, about the scholarships and funding, yeah, you can apply for the DAT scholarship. I think this is really the big one. And if you have already,
Madam,
sorry. Okay. So if you already know in supervisor, or if you already contact any supervisor beforehand, then it's much easier to get this kind of scholarship and then to come here and to do your work. So mostly the DAT scholarships works here. Any other questions?
for your
Yeah, I think I just wrote it in the chat, the scholarship name. Yeah. I don't see any other questions.
Uh, so thanks for this session, and I think everybody has got their questions answered.
No, I think I just saw one.
Okay, this is another question. No, yeah.
One more question. Yeah,
Sure,
the
yeah.
financial things during the internship. Yeah, it actually depends. Like where you are applying. Like sometimes in some internships are paid and some are not. And if you already have a that scholarship, then of course you don't have to worry about that. But actually, the supervisors here they really like this if you apply for the that scholarship and you come here for the internship, because it makes no sense that from India you come here without having any money, right? So this is doable. So I would say like you can start asking or you can check for the scholarships there, what are the requirements, and based on your interest then you can write the supervisor here or the way you want. And yeah, in case you are more interested, you can contact me. I have already given you my email ID. You can contact me separately. I can talk to my supervisor and I can then let both of you talk. Yeah. Okay. Yeah, I think now we don't have any more questions.
Yeah. So we can thank you for giving the time. Yeah, and
Welcome.
now we can end the session.
Yeah. Yeah.
Thank you.
Yeah. Bye.